my whiny little question, everyone.
Quick.
Number 47. I told you I know this.
Well, you got it wrong when
A
you did it.
low-level mutagenic agent would cause the properly controlled, inoculated, incubated, and AMES has played to have blank colonies. I think it's equally distributed, but we're not sure. The answer may be centrally located, which is
Okay, I get it. And we're talking about something on the Ames test, which we haven't covered, which is at the end of chapter 7, I believe. The Ames test is something that really did change all of science. And you can sit down, I'll go over it in just a second. The Ames test, all right, we have a very strong FDA, strongest in the world. We still complain about it, but the FDA requires that every atom of every FDA-approved drug be known, which is why I don't take supplements, because they're not regulated. You have no idea what's in them. And one of the things that they do require is, because of the Delaney Amendment that was done in the 50s, that every product put on the market in the United States has to be tested to see if it is carcinogenic. And the way in which they used to do that was large animal testing. They would, like when my dad was making a product that he sold called Treflin, that is a anti-weed. In other words, when people plant corn, they plant the corn and then they spray, while they're planting the corn, Treflin, and broadleaf weeds don't come up. If they don't come up, then you get more crop, because all the nutrients go to your corn plant, and when you're harvesting, you don't get weeds mixed in with your corn, which makes it much more efficient and everything. Well, when he was working to develop this compound to get it approved by the FDA, he had to prove it wasn't a carcinogen. And what they did was they fed it in differing amounts to 5,000 beagle puppies. And so when I was 11, I went to play with the puppies every day when he went to the lab. And one day I went back and there were no more puppies. Hundreds of runs out there with water and concrete runs, but no puppies at all. And I used to name them and pet them and come home smelling like them. And then I said to Dad, "Where are the puppies?" And he said they were sacrificed. Because what they do is they feed them for 3 years with different quantities of this, and then they anesthetize them, which means kill them, and then they autopsy them to see if there is an increased cancer rate in the tumors in the ones that got fed the compound. So a lot of animal activists and people who like puppies didn't like this. And so a man in the 1980s by the name of Ames discovered that a bacteria can do a better job. And it's called Salmonella HIS negative. And this is an auxotroph like some of you. That is a sick, twisted mutant that cannot digest or make an essential nutrient that everybody else in their genus can do. So you people that can't digest milk are sick, twisted, auxotrophic mutants. All right, so this Salmonella normally can grow and reproduce by altering another amino acid to produce this amino acid, histine. But this particular Salmonella has mutated and can no longer make its own histine. That's the basis of the entire Ames test. So, every product in the United States now is, they make agar without histine in it. Then they drill a hole in the center about the size of your pencil eraser and fill it with whatever compound they want to test. Then they streak a lawn with a Q-tip over the top containing Salmonella HIS negative bacteria. These Salmonella have a point mutation. That means one amino acid is out of order or knocked out, so they can no longer make this. If they don't make it, they don't live. So you put it in the incubator overnight, and you take it out the next day, and if there are any colonies at all, the substance is a mutagen. Any colonies means it's a mutagen because it has no HIS in this agar. Therefore, this bacteria should not grow. If the chemical causes this to flip back to normal, to revert to wild, you know, people love that, revert to wild. You didn't know that wild is natural. So when you go wild, that's natural, right? So if the colonies are near where it was, that means it is a mutagen at high concentration only. If they are distal as well as central, then it is a mutagen at any concentration. So you should be able to answer the question.
A low-level mutagenic agent would cause the properly controlled, inoculated, and incubated test.
So what would it be?
Distally.
All over,
Equally, huh?
equally.
Yeah, I got it right, and I guessed it. I didn't know the topic, I just
Now,
guessed
what's
it.
interesting
I
about
Googled
this
it, I
is
Googled it.
large animal testing only looks for carcinogens. To get cancer, you have to have a mutation of DNA. So yes, it does detect mutagens, but only carcinogenic mutagens, while the Ames test detects any mutation. So which would you rather get rid of if you were God? I'm going to get rid of carcinogens or mutagens? Which?
Mutagens.
Right, mutagens gets rid of things like causes this. This is a mutation. It's not cancer, trust me. But I would rather not have this mutation. So the Ames test is better. Why is it better? Puppies don't have to die. It gets rid of mutagens. And it's overnight with billions of bacteria. Remember generalization? The more organisms you do an experiment with, the better the results. So it's 15 cents. How long does it take? How much money do you think it takes for 5,000 puppies for 3 years? Yes, sir.
So Salmonella, it has lost the ability to synthesize which...
You're going to have to read it louder.
Salmonella has lost the ability to synthesize which one of these?
What number?
46
is rating.
What is
Lost the ability to synthesize what?
Arginine, threonine,
Okay.
tryptophan, carcinogen, one of these.
What number is this?
And what's the
46.
question part of it? 46.
It says the Ames test utilizes a strain of bacterium, Salmonella, that has lost the ability to synthesize
So it's histine, right?
Histine.
Histidine, none of these.
Yes.
Yeah, it's a fake. I put something that looked like histine. But histidine is not histine.
I have number 48. I have number
46.
Read it loudly.
Number 48. What is the what is different about the pentose phosphate shunt?
It starts with a 5-carbon sugar and takes the place of glycolysis. But it still gives you 2-pyruvate and 2 ATP.
Number 48.
48.
So the options are, it starts with glucose A, B is it starts with the 5-carbon sugar, C is it ends up with pyruvate and ATP, and D is it results in a 3-carbon compound, and E is none of these is correct.
None of these is incorrect.
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 Okay.
So it's E.
So, yeah, it's E because it does glycolysis and a 5-carbon sugar at the same time.
I have a question. Number
Won't be on there because I haven't covered it in your class,
Yeah,
but I
okay.
will lecture over it.
number 51. Which of the list of ways of moving DNA can move the most DNA?
Conjugation moves 51.
Okay, well, that's just the whole thing. Conjugation's not even part of the.
Okay, then what's the next choice?
Transformation, 1. Electroporation, I don't even have that in my notes. Specialized
Electroporation is artificial
transduction,
transformation. They can move 21.
21. Okay.
So it's B?
And then specialized transduction, generalized transduction, all of these move about the same amount of DNA.
So it's artificial transformation.
Artificial.
Artificial transformation is called electroporation. That's where you knock a hole in a plasma membrane with electricity, and you can put in really huge numbers of genes in there.
You said electroporation moves 21?
No. Yeah, 21 is the most they've done so far, but eventually they'll be able to do even more.
And conjugation moves.
Electroporation, which is artificial transformation. Remember, what is transformation? When a cell dies, DNA is released into the liquid. A nearby relative can absorb that DNA through the plasma membrane. Naturally, only one gene moves that way. But we hid behind the door, like a pervert. You know what a lurker is, right? Someone that's in your chat room but says nothing and watches all that goes on. So that's what scientists did. They watched transformation happening in nature, bacteria dying, releasing their DNA into the liquid, nearby bacteria absorbing it through the plasma membrane, and they discovered two things. Only one gene is moved, and to get it through the plasma membrane you need a protein called competence factor, which is really hard to get and make to absorb DNA through the plasma membrane. So the scientists said, well, the only hard thing is getting through the plasma membrane, and they were American. What do we do when we have a hard time getting into something? We blow it up. Isn't that what we do? You know? Do we surround people that have hostages or hiding in a house threatening to kill themselves and cut off the electricity and the water like they do in Europe and just sit there and wait until they starve? No, we go in shooting. So that's what we did. We said the only problem with getting a gene in a cell is getting it through the plasma membrane, and competence factor only will carry one, and we want more than one, so let's blow a hole in the plasma membrane. So we drop the little bacteria in saline solution and run an electric charge through it and blows holes in it, then you can put 21 genes in there. It's called artificial transformation or electroporation, electricity to make a hole. Other questions? Yeah.
166.
166.
Yes. And enterobacteria utilizes the blank pathway.
Enterobacteriaceae or Enterobacter uses what pathway?
Yeah, uses blank pathway. Homolactic acid.
2,3-butadiol or acetylene. Is that there?
No.
Acetylene.
Oh, acetylene is, yeah, sorry.
Okay. Next question. Number 7. Why don't I read it? Number 7. Protein synthesis in prokaryotes and eukaryotes has many similarities and a few differences. Which of the following is not a difference? Okay, so that means we're looking for not. All right? There is no nuclear membrane for processing introns in prokaryotes. That is true. That is a difference. B. Transcription and translation happen almost simultaneously in prokaryotes. That's also a difference. Number C. Prokaryotic 70S ribosomes do not provide post-translational modifications. That is also a difference. D. Tertiary properties of the protein are similar in both. Who cares? Tertiary doesn't really affect anything. And E, protein synthesis starts with methionine. It doesn't. Remember, bacteria, eukaryotes start with a different amino acids. Eubacteria starts with a different amino acid than archaea and eukaryotes.
So the answer is E.
E. Other questions?
I have one. Number 17.
Let me just use your paper. Sorry. Hopefully you could get the answer and write it down. Excision repair is called dark repair. In other words, there are 3 repair mechanisms for damaging DNA with UV light. Who can tell me what UV light, how does it cause damage to cell DNA?
Not all the biological molecules.
Right? What does it do? It causes two thymines to bind together instead of a cross. So it's called making pyrimidine or thymine dimers. That's what UV light does to damage your cells. Now there are 3 cell repair mechanisms. One is called photolase, and it requires UV light to repair UV light damage, which is hilarious. And it uses one enzyme, and all it does is pop a zip. It reads through, here's the slide, and it goes pop, and then it keeps going. And that's the best one. The second one is called dark repair or excision repair, and what it does is the repair enzyme reads the DNA, and it finds the damage, it backs up one, cuts it on the other end, throws it away, and resynthesizes according to the other side. And that's called dark repair or excision, which means cutting. And the third one is useless. It's called SOS repair. And that's when you've irradiated a bacteria overnight with a lot of UV light, and it has hundreds of thousands of dimers. So what does the bacteria do? It comes up and it sees where the region of the DNA where it has the most, and it cuts the whole DNA molecule and throws it away and glues the two ends together, which is, of course, useless, and it dies anyway.
Which one's light?
The first one, photolase, light repair. Next question.
148.
148. Which method of gene transfer is most significant to the new science of recombinant DNA technology as an idea? And the answer, let's see. Which of the gene transfers is most to the new science of recombinant DNA as an idea? Transduction uses viruses. Transposition, not a method. Transformation, nope. So none of these is correct. The best answer for that one would be conjugation or plasmid.
What question? 148.
The chapter on, that is mutation, 7. Won't be on there, but it'll be on the next one.
It won't be on or something? We don't know anything about
I don't care, you asked.
Oh, I have a question on number 19.
Quiet!
Metabolism is regulated so that cells may A, make only what is needed, B, conserve ATP, C, make what is required, or D, none of these is incorrect. I put B.
Incorrect.
Yes, sorry, none of these is incorrect. I
Right.
put B, conserve
Why
ATP.
do why do cells regulate ATP? Because like anaerobes get only like 8 to 12 ATP for every glucose, so they can't afford to waste any. So they only make what's needed. They don't want to waste any ATP, so all of those, none of these are incorrect. It's the best answer on that one.
No, B.
Okay.
So, I mean, I can ask this question, but the lac acron is not going to be on this exam?
Not on this one.
Okay.
Not on the one Monday and Tuesday.
Not on the one Monday and Tuesday, but the one following. So I'll ask it anyway. So this is number 23. When considering an inducible operon, offing the off signal is accomplished by...
Lactose.
I know it's lactose, but that's not what I'm
Okay.
saying. The repressor, the promoter, the substrate, the operator, or the regulator.
The substrate.
repressor. Lactose is the substrate?
It is a substrate.
Oh.
Who else? Yes?
Okay, it's actually 104.
Okay.
Which of the following genera should be utilized in the MTReview2 experiment if one expects to obtain an accurate identity? A. Pseudomonas, B. Macera, C. Aeromonas, D. Enterobacter.
It's by the MTReview2.
MTReview2.
Okay, MTReview2 needs gram-negative, oxidase-negative rods. So, gram-negative, oxidase-negative rods, and if you look at the oxidase test and the MTReview2 test, it says that there are gram-negative, oxidase-positive rods are alkalogenes and Pseudomonas. So, Neither one of those go in the enteral tube, they go in the oxyferm. Does that answer the question?
It's Enterobacter.
Okay, so it's not one of those, so it's Enterobacter. What were the choices?
Pseudomonas, Neisseria, Aeromonas, Enterobacter.
Okay, Neisseria is oxidase positive.
Or none of these should be. An option is none of these.
This is a coxy, and it's oxidase positive, so it doesn't go in the oxyferm, goes in the oxyferm, not the enteral tube. And the other one was Aeromonas,
Aeromonas,
and what was the other choice?
Enterobacter.
Enterobacter. So Enterobacter is gram-negative, oxidase-negative rod, so it goes in the enteral tube. Aeromonas we don't have as an unknown.
So that is,
These are for the oxyferm. So if they're for the oxyferm, they're not for the enteral tube. Other questions? Yep. What number? 27. When discussing the means of enzymatic control, which of the following is most related to competitive inhibition? All right, competitive inhibition is where something looks like the substrate, shaped like the substrate, will fit into the enzyme as if it is a substrate, but won't work. And that is an analog, and most analogs are antiviral drugs. They are fake DNA or RNA bases that look like AZT. It looks like thymine, shaped like thymine, will be utilized by a cell as if it was thymine, but won't work. That's an analog, which is a competitive inhibitor. Next.
I just have a general question. Since there's a big chunk of the information skipped for our lecture, can we assume that we're going to have, because we're still going to have 200 questions, right?
Right.
So what are you going to make up for that?
Mostly the card.
Mostly cards. So
Chapter
more card
4
questions.
and Chapter 9 on taxonomy.
No intro?
Oh yeah, lots of intro.
How about chapter 5?
What?
Chapter 5.
Chapter 5, some of you had it, part of it, and some of you didn't. So if you had part of it, I'm making two tests. I'm making the ones that didn't get Chapter 5 and the ones that did. Other questions?
Number 42, it's the least efficient indirect method of metabolic control is known as, and you have
Okay,
A,
let me
feedback
read it.
inhibition, B, attenuation, C, enzyme induction, D, end product inhibition, and E, catabolic repression.
And it was which, number 42?
42.
Okay, the least efficient indirect method of metabolic control. All right, indirect is those genetic ones. And they are covered in the lecture in order of least efficiency to most efficient. So the least efficient would be the lac operon induction, inducible operon. The second least efficient would be the one that is the trip operon. The next one would be attenuation. That's attenuation. And then the most efficient would be catabolic repression.
Okay.
So of these, feedback inhibition isn't even genetic.
No.
So we'd have to pick between B, attenuation, enzyme induction, end product inhibition, which is feedback again. So you only have to choose between B, C, and E. And the most efficient of all is catabolic repression. You don't spend any ATP to make lactose if glucose is there.
the question is the least efficient.
So the least efficient, that'd be the best, and the least would be enzyme induction.
Okay, so I was right. Okay, I just
And
wanted-
followed by attenuation, which is followed by catabolic repression.
Okay.
So whoever put it here has it right.
Number 147.
Read it.
During bacterial
147.
During bacterial transduction, what carries the genetic material from one cell to another?
Okay, during bacterial transduction, what carries the genetic material? A virus. So Look for any virus or look for bacteriophage or phage. Which one's there?
Bacteriophage.
Viruses.
Is bacteria there or bacteria,
bacteria.
virus
It
or
says
phage?
there's bacteriophage there as well.
So is there an,
There's a all but answered B, which is a plasmid. There's a pili, bacteriophage, virus.
highlight don't carry it? 147. Okay, 147. What carries the genetic material? Pili is wrong, bacteriophage is right, a virus is right, plasmid is wrong.
So it would be bacteriophage,
So,
right? The first
oh,
one?
I see. Yeah, that's a misprint. Highlight is not involved in transduction, so it should be all of these except A and D. You might want to correct that.
What should the answer be?
A and D.
So all of these.
All of these except A and D. Yes? Anybody else? What?
What you just said.
All of these except A and D should be the correct answer. Highlight is not involved in transduction. Yeah,
Oh, it's 146.
okay.
What molecule is released during a certain stage of bacterial growth that facilitates the uptake of DNA between closely related microbes? A. mRNA, B. tRNA, C. peptidoglycan, D. confidence factor, E. pilus protein.
Confidence factor. Remember I said in transformation, in natural transformation, one of them has to make a protein that allows DNA to pass through the plasma membrane, and that is called confidence
For
factor.
some reason, I was hearing confidence factor.
Oh, well, that's probably, they probably need confidence to get through either
Yeah.
also.
Okay, I have number 32. The enzyme used to break down glucose are usually classified as.
Constitutive. Number was?
Number 32.
32.
The enzyme used to break down glucose are usually classified as constitutive.
Yes. Glucose is the universal food, so it's the enzymes to break it down are part of the cell membrane, and that means they're constitutive, used all the time, every day. Don't have to be synthesized. Next. 161. A vitamin is a substance needed by some enzymes in order to be complete and functioning. Vitamins are used up in these chemical reactions and are known as blank because they attach at the blank. They are known as coenzymes, and they attach at the active site. So let's see if we can get there. Cofactors, active site. So A would be the right answer because it is a type of, coenzyme is a type of cofactor. It would be better if the answer was cofactor and active site, but I don't think it's there.
That's what I had, yeah, cofactor and.
How about C? How about
C?
C?
I thought mineral
Coenzyme active site's the best answer, yeah.
Wait, I thought you said cofactor was better.
Cofactor active
Cofactor,
site.
a coenzyme is a type of cofactor. So if you're getting more detail, that's a better answer.
So A is.
A is true, but the better answer is C, right? That's why people hate my test, because you have to know something, you have to make a judgment.
But you just said that cofactor was better.
No, I said that coenzyme is better than cofactor because both minerals and vitamins are cofactors. But coenzymes are more specific. A vitamin is a coenzyme.
Okay.
It is a type of cofactor. There are two kinds of cofactors, minerals and coenzymes, which are vitamins.
But we didn't do that. We didn't go.
Right.
Okay.
One of the other groups did do that chapter.
I mean, we went over it, but I wouldn't have known that from your notes.
Well, I mean, you know, I did go over it. Well, you will, actually, your class will see the little video that explains that both of these are called cofactors. One of them is a inorganic mineral cofactor, and the other one is an organic coenzyme cofactor. Yeah.
177. A positive result on the blank test results in the production of a keto acid. The choices are A, iron test, B, OMPG, C, citrate, D, tributylin, D, PA, E, VP.
Well, I'll tell you the truth, I think I forgot. What number was it? What number?
177.
Okay, a keto acid, citrate.
Citrate.
If you look up citrate in the unknown test, which we can do right now. I believe it says acetic acid.
It's phenylalanine.
Nope, you're right, it is phenylalanine. Just, let me just get to it.
Which test uses Vaseline for its preparation? It's not there. It's
Okay.
Oh, motility, yeah.
I believe the word keto acid is
No,
here.
175 on the website. Yeah. LP2.
Yep, see there it is, right there.
Yeah.
See whether or not phenylalanine deaminase enzyme are able to deaminate phenylalanine to a keto acid.
So it's on our
I
cards.
told you I fooled myself on that one. Frightening. Yes.
Number 9, a blind culture is a culture that maintains the organisms with the characteristics as originally defined.
Reference. He wants to know, one of the questions is, where is all the cultures as they are in the reference books kept? What's the place where they're all kept? It's kept at the American Tissue Culture Collection in Chicago, and those cultures that you can order are called reference cultures. They are the standard E. coli or whatever.
So what would stock culture be?
Number 9.
All right, what was that other mumbling in the background?
I said, what would a stock culture be then?
Stock culture is the backup
Reference
used in
right
your
there. Number C.
experiment. 158. 158. Which phase of the growth curve is utilized to measure all published data? Log growth. Or exponential growth. What number was it? 158. On the growth curve in chapter 6, it says that the second level of growth curve, remember everything in life has a growth curve. And when you're dropped into optimum conditions, you are in lag, a period of adjustment. Then they will reproduce as fast as possible and metabolize as fast as possible. 1 becomes 2, 2 becomes 4, 4 becomes 16, and that's called log growth. When the waste builds up and there is no more food to eat, the number being reproduced equal the number dying, and once all the food is gone and all around you is waste products and there is no food and you're super crowded, they will die at an exponential rate. Here to here is when involution occurs. Somewhere in stationary and always during death, involution or upside down occurs. And this is why I told a whole bunch of people, when you do your first gram stain, and I say it's the size and shape that matters, and people go, you told me mine was gram negative, and yet I see purple. And the reason you see purple is it was in stationary or death phase, because log ends at 48 hours. So if your culture is more than 48 hours old, it will be upside down on results, which is involution. That's why we take all inoculations that are used for reference book results are always taken in log growth. So this is 2 hours long, this is 2 hours to 48 hours, and we leave ours 12 hours in the incubator, and we artificially maintain log growth by refrigeration. And what's our limit on refrigeration? How long can you keep yours in the refrigerator?
24 hours.
Nope.
Oh, 12 hours.
No, 12 hours is in the incubator. It's growing.
48 hours.
This is when you put it in stasis. No, that's in the incubator. 10 to 14 days. Every 10 to 14 days you're supposed to make a new slant because refrigerators dehydrate. leave your Chinese food rice in the refrigerator with the top off for 2 days and come back, you'll have concrete.
So the
They
answer
dehydrate.
to that is none of these are correct because the
You're
options
memorizing?
It's
are
growth
lag
phase.
phase. There's no log growth here.
Yeah,
Okay.
so it's.
All you need to know is what is the correct answer. Remember, if you memorize the question, it won't be there.
The correct answer is log.
You have to understand it.
So you wouldn't say that it's growth phase?
It's growth phase.
Is it correct
Huh?
answer?
Yeah, it's growth, log growth,
It doesn't say
exponential
log, it
growth.
says growth phase. That's
I know.
not right.
This is called log growth or log exponential.
And is that when they multiply the most?
Yes,
The log growth.
and this is where
It would be.
what they call generation time is defined. How long does it take one to become two? It's always defined in log. Every published piece of data in your Burgess manuals is assumed to be with an inoculum from log growth. If you took an inoculation, like some of you put your tubes back in the incubator, your stock and your working, and put them in the incubator, which means they were in here when you, and you could not use them to inoculate your test because when they're in stationary or death, acids that have built up have eaten holes in the plasma membranes and cell walls, and so the bacterial results are unreliable on any test. Any further questions? We have 9 more minutes.
Are we gonna switch one over to word problem?
No?
197.
Julio, you have a phone call?
Yeah.
Okay, so 197. This is from chapter 7 about mutation, and you're going to get it eventually anyway, so there's no reason why we shouldn't go over it. Mutations in DNA are almost invariably important and consequential. The answer is no. Overwhelmingly, remember, a protein is 120,000 triplet codes. That's the smallest protein we've ever discovered, 120,000. You get one of those wrong, is it going to make a difference? No. It's still going to fold into the right shape and still function. So the only time a mutation is super important is if it's frame shift, meaning it knocks out a base and moves everything down, or it's in an initiator or terminator. Because remember, you don't want fingernails growing out your asshole. Alright, so the answer is definitely, usually false, as mutations are uncommon and often inconsequential because they're not genetically transmitted. That's true. Let's see what C is. False as mutations are often corrected by repair. That's also true. D, false as mutations in DNA do not matter as multiple codons. That's true also, so the correct answer is E. All of these are correct, except A.
Are we gonna finish the word problem? I think we only did
No.
2 out of the 4 or
No
5.
more time. Other questions?
156.
What is it?
How do obligate anaerobes obtain most of the
How do obligate anaerobes obtain most of their energy? It's
glycolysis, fermentation, Krebs cycle, anaerobic respiration, oxidation?
anaerobic respiration. Anaerobic respiration includes glycolysis and fermentation, and that's the only thing anaerobes can do.
Here we have 3.
What?
Glycolysis, fermentation, and anaerobic respiration.
Okay, so glycolysis is a part of anaerobic, and so is fermentation, so anaerobic includes them both, so that's the best answer.
161. The key proteins in the cell that determine its properties for identification using Burgess are known as A products, B substrates, C sugars, D catalysts, E enzymes.
Enzyme. Does your microbe contain the enzyme lipase, which enables it allows it to break down lipids as blah blah blah?
What
We're
number
always
was it?
looking for enzymes or the end of pathways, the food product or the enzyme.
149. Which method of gene transfer is most significant to the methodology of the new science gene therapy?
Okay. So the question number was?
149.
149. And the question was, which of the methods of horizontal gene transfer is most relevant to the new science of gene therapy? And that's transduction. Once they learned that a virus could carry genes, they took the genes for cystic fibrosis to make that little to digest the fluid in your lungs that you make every day, and cystic fibrosis patients can't digest that fluid, so they drowned in their own fluid. So they found genetic babies that were going to be born with cystic fibrosis. They knocked out a gene from a virus and had them breathe a recombinant virus containing the gene they were missing. It's a cold virus. It infected their respiratory system and carried the gene back to where it was defective and cured a genetic illness. And that's called gene therapy. And we discovered it by looking at viruses carrying genes, which is transduction. One more question. No more questions. So if you want to do some lab, we're doing lab in the room. I will be in a meeting at 11, but I'll be right next door, and I think Naira's already there. I'll get out the material, and we will see you on Monday or Tuesday. There. P.S. Listen. Okay. Breathe. Thank you. On Saturday night,